Since 1997, the Clinimax cell separation system has been proven for developing and establishing cellular therapies worldwide. Today, the Clinimax Prodigy brings this trusted technology together with a new range of sensor-controlled cell processing capabilities, giving you complete manufacturing workflows from cell preparation to final product. The Prodigy automates and standardizes the complete manufacturing process of cellular products. At its core is the Sentry Cult Unit, which enables complex processing and cultivation within a disposable chamber. Fully integrated centrifugation takes place with camera-controlled layer detection for cell washing, buffer exchange, and cell concentration procedures. Using these standardized manufacturing methods ensures reproducible manufacturing every time. The Prodigy eliminates the need for highly specialized handling of different appliances, greatly reducing user-to-user variability and error. The Prodigy integrates all of the required manufacturing steps into one closed, sensor-controlled system. And fewer manual steps, together with fewer cleanroom requirements, is essential to cost-efficient cellular product manufacturing. Features like the disposable tubing sets and sterile connecting ports for GMP reagents and media also ensure the highest safety. Our flexible programming suite allows full customization, empowering the translation of your ideas into GMP-compliant cellular products. Let Miltenyi Biotec help you master the complexity of cell processing. Learn more about the Clinimax Prodigy today and visit miltenybiotec.com/prodigy. The MaxQuant Taito cartridge represents the heart of the MaxQuant Taito sorter. It is a fully closed system containing 3 chambers: an input chamber, a positive sort chamber where target cells are collected, and a negative sort chamber where non-selected cells remain after each sort. Note, sorting happens exclusively in the cartridge. There are no fluidics within the instrument itself. All 3 chambers can be accessed from above through Luer lock ports, which allow for easy loading of the sample. After loading the sample into the input chamber, the cartridge is placed inside the instrument. At the beginning of the sort process, pre-filtered air coming from the instrument enters through a 0.1 micrometer filter at the bottom of the cartridge. The air then moves through an air channel and finally enters the input chamber through another 0.1 micrometer filter. The very mild air pressure drives the cell suspension through a microfluidic channel onto the microchip, where the selected target cells are then sorted into positive and negative fractions. After entering the microchannel, cells are interrogated by 3 lasers. Upon identification of a target cell, a magnetic pulse opens the microvalve, which then redirects the target cell into the positive collection chamber. The whole sorting procedure happens at a very low pressure of only 3 psi, and there is no decompression or charge applied to the cells. After the sort has been completed, the MaxQuant Taito cartridge is taken out of the instrument, and the sample can be retrieved under a sterile hood for further processing. As you can see, sorting within the closed and sterile environment of the MaxQuant Taito cartridge is much gentler and easier than on a conventional droplet sorter. MaxQuant Taito. Fast and easy, gentle to cells, fully sterile, and completely safe. Visit us at miltenybiotec.com/Taito. Miltenyi Biotec. In-process and quantity control steps during cell manufacturing are often performed by flow cytometry. In this context, Consistency and reproducibility of data are of key concern in order to provide reliable production of cellular products. Whether working in a centralized or decentralized model for your cell manufacturing process, standardization of cellular control steps by flow cytometry can be challenging. When upscaling your protocol for commercial purposes, several components influence how well you will be able to standardize your flow cytometry assays between individual instruments and across different sites. There are three main aspects that add variability to your assays: the instrument itself, the operator, and the reagents. Miltenyi Biotec offers solutions to tackle all three issues. Smart Gain technology for standardized instrument setup, Express Mode software tools for standardized experiment setup and data analysis, and the Stain Express reagents for standardized and lot-to-lot consistent cell staining. Smart Gain technology on our instruments enables you to transfer assay calibration settings from one instrument to another. The automated digital adjustments provide consistency in signal intensity throughout time and across devices. In practice, this means it doesn't matter who is running the assay, where they're running it, or even when. An identical setup is assured, and as a result, MaxQuant instruments produce comparable standardized results every time, everywhere. Express modes come in handy for assays you need to run frequently, as this software tool automates routine manual flow cytometry procedures via predetermined experiment settings to ensure operator-independent experiment setup, autonomous sample acquisition to save precious operator time, and automated gating to prevent human error. Governed by mathematical algorithms that are based on thousands of actual experimental results, Express Modes ensure superior analytical accuracy and increase experimental reproducibility. Stain Express reagents are ready-to-use antibody cocktails, carefully dried down to enable convenient storage at room temperature. These multicolor antibody panels are thoroughly validated to ensure simple and consistent cell staining during in-process and quality control steps. Unlike conventional antibody cocktails, Stain Express antibody cocktails are made from recombinant antibodies using our proprietary reaffinity technology. Recombinant engineering of these antibodies provides superior purity, eliminates the need for FCR blocking, and enables analyses with a single isotype control. This allows for the generation of high-quality data while saving effort when setting up experiments. Ready to standardize your flow cytometry assays? Contact us via our website, miltenyibiotec.com. Staggering in all its complexity and beauty, it's no wonder we can barely wait to unravel nature's mysteries. The Maxima imaging system is an easy-to-use, fully automated instrument that rewards users with ultra-high content imaging on a scale that is simply unrivaled by anything else. So intuitive that users can simply plug and play without any special training. Imaging experiments can begin at once. Powerful and dependable, the Maxima instrument helps researchers unlock new insights, making it the workhorse of any laboratory. Our multi-parameter imaging cell screen technology uses the principle of iterative staining with any of our 1,500+ validated fluorochrome conjugated antibodies to acquire microscopy data. The process comprises 3 main steps, all of which are conducted in a fully automated manner. First, samples are stained with recombinantly engineered reaffinity and readylase antibodies from our ever-growing portfolio. Next, immunofluorescence images of the desired sample areas are required. Finally, the fluorescent signal is erased either by photobleaching or by specific and controlled fluorochrome release using our sophisticated readylase antibodies. Both mechanisms can be used in the same experiment without harming sample integrity. The process then restarts and continues through as many iterations as you have antibodies to stain with. In this way, hundreds of markers can be acquired from just a single sample. Whilst the instrument runs, users can busy themselves around the lab or else analyze images in real time. The powerful QI tissue image analysis software allows for the selection and direct localization of cells based on the expression of markers. It easily handles large amounts of data, making it simple to gate cells in scatter plots, build T-SNE plots, heat maps, and much more. Sample material is so precious it makes sense to extract as much data from it as possible. The Maxima imaging platform gives you the flexibility to characterize specific cells and produce a vast, meaningful array of data, all from a single platform. What's more, the sample remains unharmed and can be used for downstream applications. The complete platform, including the instrument, validated recombinant antibodies for guaranteed reproducibility, click-and-place sample carriers, and intuitive execution and analysis software, helps you to unlock new insights and push your research further, faster. See more than ever before. The Maxima Imaging Platform. Scientists continually enter unknown territory to explore complex mysteries and eventually make stunning discoveries.
The sample chamber can host an entire cleared mouse model or allows automated imaging of multiple samples. Samples can then be imaged sequentially.
Time and again, scientists blast off in new directions, uncovering areas previously unknown to mankind.
Optimized illumination guarantees homogeneous fluorescence excitation.
New insights allow us to answer questions we didn't even know existed.
MI Plan objectives are available for 1.1, 4, and 12-fold magnification.
Scientists look at the big picture and study the details.
The instrument allows automated switching between different objectives and magnification lenses.
And then rethink their hypotheses. Off the beaten track, they blaze the trail towards new insights. Scientists dive deeper and deeper into the details and gain original perspectives where science sheds its confines and becomes an art form. Realize your commercial manufacturing vision with the Clinimax Cell Factory. At Miltenyi Biotec, we streamlined cell processing workflows by reducing complex manual handling steps. The Clinimax Cell Factory facilitates easy expansion to commercial scale production capacities. The heart of the factory is the Clinimax Prodigy platform. It transforms contemporary cell manufacturing. Profit from its flexibility in manufacturing a variety of cell types. Reduce the risk of contamination, as there are no open handling steps. Automation allows standardized processes. It minimizes hands-on time from the operator and eliminates variability of cell products. Closed system cell processing reduces clean room requirements and enables parallel manufacturing in one room. In addition to our expertise, we provide clinical-grade consumables, as well as documentation for regulatory support. It all comes from one source. In the Quality Control Suite, Automated flow cytometry is done by MaxQuant analyzers. Our manufacturing model is comprised of separate material and personnel airlocks, a Class C manufacturing clean room, and a room designated for quality control. The Max unit model is easily scalable, from a global network of manufacturing suites to a large central manufacturing facility. The Clinimax Cell Factory can grow with your vision. Overcome the commercialization challenges. Let the Clinimax Cell Factory streamline your processes and realize your goals in cell and gene therapy. Miltenyi Biotec. The Clinimax Prodigy is known for its superb automated closed system cell processing and GMP-compliant viral transduction. We've now added more freedom and flexibility to how you can perform genetic modifications on your cells. Welcome Clinimax Electroporator. When connected to the Clinimax Prodigy, this additional module integrates an electroporation step to your automated cell manufacturing protocols. The test cuvette adapter offers the possibility to optimize your protocols on a small-scale level before starting your larger cell production, saving time and resources. As the Clinimax Electroporator is controlled by the touchscreen of the Clinimax Prodigy, all settings can be adapted to your needs. For large-scale applications, cells are processed within a single-use closed tubing system. The Electroporator tubing module contains the integrated electroporation cuvette. This is where fully automated transfection takes place. Before electroporation, your cells begin their journey within the Sentry Cult unit of the Clinimax Prodigy. The rebuffered cells are then directly transferred to the Clinimax Electroporator. Cells and nucleic acid remain separated until electroporation takes place. The risk of DNA or RNA degradation is reduced, and the amount of nucleic acid required for successful electroporation is decreased. For each transfection step, predefined amounts of cells and nucleic acid pass into the electroporation cuvette. Both then enter the integrated mixing structure to ensure a homogeneous suspension between the electrodes. Now the electrical pulse is administered on the cells. This pulse creates temporary open pores in the cell membrane through which the nucleic acid can enter the cell. This entrance can be supported by a second electrophoresis pulse. The cells are now transfected with the foreign nucleic acid. Depending on the conditions, a transient or stable transfection is possible. The pulse settings can be adjusted via the user interface. The transfected cells are transferred back into the cultivation chamber of the Clinimax Prodigy, where they are suspended in recovery medium and ready for downstream applications. Profit from automated and closed system cell transfection, the winning combination of the Clinimax Prodigy and the Clinimax Electroporator delivers reliable and consistent results of your cell products. Miltenyi Biotec.